Okay, welcome everyone. Today we're going to have our uh, seminar, the JSMN seminar, and today we have Dr. Adriana San-Miguel from North Carolina State University, and she's going to talk about her interesting research about C. elegans. Uh, Adriana is an assistant professor in the Department of Chemical and Biomolecular Engineering at North Carolina State University. She's a part of synthetic and systems biology cluster that hopefully we're going to have a lot of uh, advancements in this field for the current situation that we are in. And she has received a Bachelor of Science in Chemical Engineering from the Monterrey Institute of Technology in Mexico. And she obtained a PhD in Chemical Engineering from the Georgia Tech in 2011. And after that, she was a postdoc fellow at Georgia Tech at the Dana-Farber Cancer Institute. Then she joined NC State, and now she has a very prestigious NIH K99 award that I think transferred her to current position that she has. So without further ado, I remove myself from the screen, and Adriana, floor is yours. Please start.
Okay. All right. Well, thank you so much for that introduction, and hello everybody that's connected to this broadcast. I'm very happy to talk to you today, remotely or virtually, given the circumstances. But thank you all for joining. I'm going to tell you about the work we do in my lab. As it was mentioned earlier, I'm an assistant professor of chemical engineering at NC State, and I started my lab in 2016, so I've been there for about 4 years. And so I'm going to tell you about the engineering approaches we use to study different biological processes in a model organism that's called C. elegans. And so I'm going to start with a very broad background about why we're interested in doing this. Right now, as you all probably know, the amount of data we have in terms of molecular information is huge. So right now, because of sequencing, high-throughput sequencing, and next generation sequencing, we can very easily get genomic information. Although initially it took a very long time to get, for example, the genome of the human, now we can get this information really quickly. And so in biological research, getting DNA sequences and RNA information is relatively simple. Now, on the other end of the spectrum, which is the organismal characteristics, that's where we have a lot fewer data. And really the goal of biological studies is to connect these two, right? How do we connect the information that we have at the genomic level with the characteristics at the organismal level? How do we figure out what are the functions of genes? How to figure out how they all work together to drive function? And what happens with disease or with aging? What goes wrong? And how do we connect the two? Now, in order to understand this connection between genes and characteristics or biological processes, we usually use model organisms. The reason for that is that even though we would like to figure all of this out in humans, that's really difficult to do because humans take a really long time to grow, and also it's just unethical, right, to do all these experiments in humans. And so we use model organisms, and there's a wide variety of model organisms. We have some that are very low complexity single cell organisms, for example yeast or single cell cultures, and there are some more complex organisms such as mice or monkeys. And usually these two characteristics, the ease of culture and the complexity, are anti-correlated or inversely correlated, I should say. So for example, doing experiments with yeast is faster. They grow really fast. You can get thousands or millions of cells really easily, and that means you can probe the function of many genes or see the effect of many drugs in these cells really quickly. Now, if you go higher complexity, for example, if you do work with mice or monkeys, those experiments take a lot longer, and you would have a lot less numbers in the total number of individuals you have. And so because of this reason, experimentation in all of these models is important, because as you go higher in complexity, you're really getting closer to what a human would be. They're closer to human, but you also can do experiments in such high throughput. And so the model organism that we use in my lab is C. elegans, which is highlighted here. I'm going to actually go to presentation mode because I don't think I'm in presentation mode. There we are. And so we use the model organism C. elegans. So let me first tell you about C. elegans. So C. elegans is a simple multicellular organism. It's one of the simplest organisms that we have that we used to study biology, but that's multicellular. It's a little worm that lives in the soil and the rotting fruit. And it's become very popular in the past couple of decades as a model organism for multiple reasons. One of them is that they're really easy to culture, so you can grow hundreds and thousands of these animals really quickly. One single animal can produce anywhere from 200 to 300 progeny. They have a really short cycle, so from the time they're laid as eggs until the time they're able to lay eggs themselves, it's just about 3 days. Their genome was one of the first ones to be fully sequenced, so we know that information. Also, very interestingly, C. elegans has what's called determined cell lineage. And what that means is that all the animals have exactly the same number of cells, and they are all organized in the exact same way. And the reason that becomes very useful is because we know that all of these little worms have 302 neurons. And back in the day, a couple of decades ago, somebody went in and sliced all of these, or took little samples of these animals and did a lot of work in electron microscopy to reconstruct how each of these neurons connect to each other. And that's what we call the map neuronal wiring. We know exactly which neuron connects to which is really, really useful for neuroscience studies. This little worm is also transparent, as you can see here, and that becomes important because now with the use of fluorescent proteins, that means we can just look through the tissues of this animal without having to section it or fix it or do anything else. And finally, we can see the processes, biological processes, as they occur in a living system, because we, as I said, we don't need to fix them. We don't need to sacrifice the animals to see through the tissues for any specific process we're labeling with a fluorescent marker. Okay. Now, in C. elegans, usually we can have different types of readouts. And so I'm going to first tell you about some simple readouts. And so here at the top, you can see 2 sets of animals. And this comes from a paper from PNAS a little over 15 years ago. But what you see on the left-hand side are animals that have sort of a wavy shape. And on the right-hand side, you have animals that are completely straight, right? And that tells you that the animals on the left are alive and the animals on the right are dead. So that's something very easy to see. If you were doing an experiment where, say, you're trying the function of a drug, or where you're trying what happens when you mutate one specific gene, and the readout that you're looking at is alive or dead, that's very easy to see just visually, right? Or to determine. Other very simple readouts include animals moving or not moving, or whether an animal is large or small. Now, today, we have some more complex readouts that are a little bit more difficult to analyze. And here on the right-hand side, I have a few examples, or I have one specific example. And here on these images that come from fluorescent microscopy, what you see are a row of dots, right? So you see a top box that says wild type and a bottom box that says C2. I'm gonna try to see if I can show a laser pointer. There we go. C2. And each of these dots is a synaptic connection. So that's a connection between a neuron and a muscle. And each row of this is a different animal. So we have 4 samples for the wild type animals, or just normal animals. And at the bottom, we have 4 samples for animals that have a mutation in a gene called C2. Now you can imagine that characterizing these patterns of connectivity is a lot more complicated than saying a worm is alive or dead. But these are still things that we're interested in. And some other things we're interested in looking at are things like protein aggregation, or the structure of neurons, or neurodegeneration. And these are difficult readouts to quantify. And so in order to address these problems of how to characterize these more complex phenotypes, we have developed platforms that allow us to do high throughput experimentation and quantitative analysis in C. elegans. And so basically the tools we use involve 4 main things. The first one is microfluidics, and I'm going to tell you about all of these after this slide. The first one is microfluidics. The second one is image processing. The third one is some external or custom-built hardware. And the fourth one is automation and control. And so what we're doing with these systems is we're developing an integrated platform that allows us to do automated experiments so that we don't have to individually go in and pick animals and immobilize them, for example, and put them in the microscope. And then the second one is we also integrate here image processing, which is a big part of what we do. And what that allows is to go from images that looks like the ones I just showed you, let me just quickly go back like this, to get actual quantitative information of what this image tells me. For example, the number of dots, how far apart they are from each other, things like that. Okay, so let me tell you about each of the components of these integrated platforms. The first one is microfluidics, and some of you might be familiar with microfluidics. This is a technique that's become very prevalent in many scientific fields, and it was developed a couple of decades ago. And so in microfluidics, basically what we do is we develop these very small chips. Here you can get a sense of scale, where we can generate these very, very small channels on the micron scale. And so all of these channels you see here in this little device, all of the colored regions are channels. And these channels can be anywhere from, I mean, it depends on the technique used for microfluidic devices, but the ones we use at least can range from, say, 5 to 10 microns all the way to 500 microns is the scale of these little channels. And the animals we use, I forgot to mention this, the animals that we use are about 50 microns wide and about 1 millimeter long. And so these microfluidic devices actually help us in working with C. elegans because they enable high throughput handling of these worms. If we do not use microfluidic devices, what that means is we need to go in with a little wire and pick these animals one by hand. Imagine these animals are really, really tiny, so it's very difficult. In addition, even if we do this by hand, that means we would have very low sample numbers because that's very time consuming. So these microfluidic devices allow us to handle that, put these animals in these little channels, and we can handle them a lot easier because we use fluid pressure to drive them from one chamber to the other one. And it also means that we can present very well-controlled environmental conditions. For example, if we're interested in the response of the animal to a specific concentration of a chemical, we can do that very easily in these microfluidic devices because we can just inject the sample that we want. Whereas if we use traditional plates, agar plates that we use to grow these animals, then all environmental conditions are a lot more difficult to control. And so just to give you an example, this specific device that I'm showing you here, it's what we call a worm sorting chip. And so in this device, what we're doing is we are looking through animals and sorting them based on their characteristics. So as you can see here, we have 3 different colored channels. The red channels is what we call the flow channels. That's where our animals flow through. The green channels are valves, and then the blue one is a cooling channel. And let me explain how this works. So in this worm sorting device, basically we inject animals through this inlet right here. Then animals flow to the right, and then they reach this really narrow horizontal channel you see here. And in this little channel, we trap our animals, and I'll tell you how we trap them in a little while. We trap our animals here, and that allows us to take an image with a microscope. Then we do some analysis on the computer using MATLAB to determine whether an animal looks normal or not, or whether we're interested in sorting it, for example. And based on that, we can open or close the appropriate valves to route our animal to one outlet or to the other outlet. And then we can collect these through tubing that leaves the device. Now, how does the valves work? How do valves work? So these valves are channels that do not have an outlet. And so what happens is these little circles you see here, that's where we inject fluid. So we inject fluid here, for example, but the fluid has nowhere to go because there's no outlet. And so what happens is we pressurize these channels. And since the material that this device is made of is PDMS, which is a clear, flexible polymer, when we pressurize these channels, they will basically pinch the channel next to it because the channel will expand. And so that's how we can use these little channels as valves, as pinch valves. And we can trap our animals, open and close valves as we want with this device. And so let me just give you an example of how this specific worm sorting chip works for an imaging cycle. And what you see here at the bottom is just this little section right here, which is the imaging region of this device. So basically at the beginning, all of these blue, solid blue regions are valves. And you see this valve is closed, this valve is closed, and these two are open. And so at the beginning, an animal comes in, and then we close the inlet valve. Then we do our analysis. We take our picture of whatever specific feature we're looking at. MATLAB processes this image and determines whether this animal is something we want to isolate or not. And then the animal is routed to one of the exits. We did that by opening this valve and opening this valve. We could also route it this way, in which case we would have to close this valve and open this valve. And so that's basically how it works. And I'll tell you more about this sorting chip in a little bit. But now let me move on to the second main component of these high throughput platforms, and that is image processing. So basically, now that we have this microfluidic device and that we can image many, many animals at once or sort them, what happens is we end up with a lot of images, which is also a lot of data. And in addition to having a lot of images, we also have images that have difficult to analyze characteristics. So in this example, I'm going back to this image I've shown you before, where we have this row of little dots, and those dots are synaptic connections. And so if we want to quantify things like how many connections this worm has, or how big these connections are, so how big the little dots are, doing this for many images is difficult. And so the way we do this is we take a two-step approach. And the first step is image segmentation. And some of you might be familiar with this, but basically what that means is we go from a raw image like this to a binary image. And so this binary image is just zeros or black everywhere where there are no synaptic connections, and white dots or ones where there actually are objects that we identified that are relevant. We can do this through machine learning or through some traditional image processing approaches called thresholding. And then once we have this image, it's fairly easy to write algorithms in MATLAB to extract any specific characteristics we're interested in. For example, then again, the number of dots, how far apart they are from each other, etc. So that's the second part. The third part is external hardware that we use to actuate the device. And so what you see here, this is a pressure box that we built. And basically that's a pneumatic source. It has compressed air, and each of these little outlets that connects to our device is basically injecting pressurized air. The other thing we can do with this is inject pressurized air to a tube that has the stock solution. And then from that pressurized tubing, from the pressurized reservoir, we can inject fluid into our microfluidic device. So we use this box both to open and close valves and also to pressurize sources of fluid that come into our device. The other thing we do is we use MATLAB to control what this pressure box does, so that in that way we can, through the computer, open and close valves that are in our device. We also have some heat exchangers to reduce the temperature in our system, and I didn't mention this before when I was showing you the microfluidic device, but basically we can cool down our system using this cooling channel by injecting fluid that's at low temperature. And this low temperature happens through a heat exchanger that we build ourselves using a Peltier module. And we want to control temperature because if we want animals to stay still to take pictures, one way we can do this is by cooling them down. Okay, and then last thing we do is automation. As I said, we use MATLAB to control what the device does by controlling itself, the pressure box. But MATLAB can also control, and we're using MATLAB, we can use other software, but MATLAB will also control anything that's happening in the microscope. And so here's just a screenshot of a graphical user interface we've built that enables us to control everything that's happening in our experiment in our microfluidic device. It allows us to open and close valves, to take images or Z-stacks, which are images at different focal planes. And it allows us to do screening or sorting animals based on specific rules that we set. And I'll come back to this automation in a little bit when I'll tell you about genetic screens. Okay, so basically with these platforms that we integrate, what happens now is we're able to get large sample numbers, and we also are able to get quantitative characterizations to go from raw images to quantitative analysis. And that means we are now able to really look at biological questions that require analysis of complex readouts. So rather than looking at alive or dead, we can now say, I'm going to look at readouts such as the number of synaptic connections or other things like neurodegenerated neurons, for example. So we can look at subtle changes, we can look at small and dim structures that are difficult to quantify, or noisy and heterogeneous readouts. And so this is sort of the big introduction of what my lab works on. I'm going to now tell you about 3 stories of how we're using these tools for specific biological questions. And so the first one is how we use microfluidics and machine learning to study aging of the nervous system. The second one is how we use microfluidics for genetic screening. And the third one is another project we're using to try to predict longevity using CRISPR and image processing. Okay, so the first story I'm going to tell you about is about how we study aging of the nervous system. And so first, let me show you why we're interested in that. Here on the right-hand side, what you see are, again, these little synaptic connections that are going to keep showing up in my presentation because that's something that we're very interested in. And so what you see here is these are connections from one single neuron called DA9 onto muscles. So it's a single neuron and it makes all of these connections. And as you see, I mean, this would be hard to tell if it's the first time you see images like this, but at day 3, they're all really well aligned and organized, and they all have approximately the same size. By day 12, they are all a little bit disorganized, and they're not as well aligned, and their shape has grown, but they're not homogeneous in size anymore. So we've seen this, and then there are also reports out there that show that the nervous system changes morphology with aging. And so here on the left-hand side, we have an example from this paper where this neuron that looks like this at day 2, by day 16, it starts to create these sort of branches, right? And then here on the right-hand side, we have a nerve at day 3, and then at day 16, it starts to break up or create these little bubbles. And so we're interested in two things characterizing these morphological changes in order to eventually say, how do these morphological changes relate to functional decline of the nervous system? And so before I get into how we're doing that, let me tell you about a big challenge in doing this type of analysis. And so the first challenge we have is age synchronization since the elegans. So you can imagine that if you want to do an aging study, for example, say, how do neurons look at day 15 or day 20 of this animal's life? By the way, animals live about 3 weeks. And so the question is, how do we keep a population that's age synchronized? Because when you're taking these measurements in the microscope, you want to know how old your animals are, right? And so let me show you why this is difficult. So when we have these little animals on a plate here, I'm just showing you the initial population. We have a few animals in blue. And then let's say at day 2, they all start to reproduce and lay eggs, right? Then you have a bunch of little eggs on the plate. By day 3, all of these eggs have hatched. And so now you have the original population and a bunch of new animals that initially are smaller, but at some point, say day 3 or 4, say day 4, they all look exactly the same. Here we can distinguish them by color, but in reality, they all look exactly the same. And so this starts to get worse and worse as time goes by. You end up with a population that has mixed ages. And so that's really not useful to do aging studies. Traditionally, what people have done is they've used chemical sterilization. So what they do is they put a chemical in the plate here that prevents animals from reproducing. So you never reach this problem. You will only have blue worms, right? Because they will never lay eggs. This chemical people use is called FUDR. And FUDR is a chemical that's used in chemotherapy. So it prevents DNA synthesis, and it's a very drastic change to perform on worms, because it's been shown that that reproduction is directly linked with longevity. And so to solve this problem, the first thing we did, my student Sahan Saberi, who is one of my senior grad students in my lab, he developed this really interesting tool, which is a microfluidic device, as you can see here. It has 2 chambers. On the left-hand side, we have what's called the culture chamber, and on the right-hand side, we have the imaging channels. And here you have the design actually on the right-hand side. So in the culture chamber, what happens is we have all of our animals growing, and this culture chamber on the sides have these little narrow channels. So it's really like a filter, because in these little narrow channels, what happens is a few of the progeny or eggs can leave through these channels. And so we just keep our population here. We let them lay eggs, let them reproduce, but we're filtering out all the progeny, all the babies. And so we are left only with a parent population. And then these animals are brought back to this second side, which is the imaging channels, because we want to do periodic imaging to monitor how the nervous system is aging. So we send them to these imaging channels on the right-hand side, and then we can put them in these little channels, straighten them up, and take pictures in the microscope. Okay, and so then using this approach is how my student was able to characterize aging. Specifically, he was looking again at the synaptic pattern. So we were interested in seeing what happens. Does the number change? Does the size change, etc.? And so what he did is he took an image processing approach to go from images like this to quantitative analysis. And what you see here at the bottom is 61 metrics he was able to quantify from these. So these metrics include, here's a couple of examples: the mean size of the largest half of the puncta, so how big the largest little dots are, what's the mean distance between these dots, and other things like that. And here we have also the number of beats, the intensity, the size, and the density. And you see these things change through aging. In the bottom, this heat map is showing how drastically these features have changed as compared to young animals. So the more blue, so if you go from top to bottom, you basically are aging. Aging goes from top to bottom, and the more blue this graph becomes, is the more drastic the changes for those. So that was a method that he used to track the synaptic aging. And then after that, we were interested in a different problem in aging of the nervous system. And so now we got through chats with a collaborator, colleague at Duke University, Professor Dong Hyun, we got interested in this other phenomenon of aging of the nervous system. And let me first tell you what this is. So here's a picture of an animal, of a C. elegans worm. And these little structures in green represent this neuron called PBD. So this is one single neuron. Its role is to sense touch. That's why it has all these terminals throughout the entire body. And what you see here is that in young animals, they have really nice, well-structured dendrites or terminals. But when they get old, two things happen. One is there's no longer this nice organization in the dendrites. And the other one is that there is an appearance of beads. We call these beads, but you see all these little bubbles that show up. And so, as you can imagine, making sense of these changes or quantifying them in a way that can allow us to track what's happening is very difficult with just visual inspection. And so this is a collaboration with Dr. Kevin Flores in the biomath department at NC State, and that was also work that my student Sahan Saveri did. And so what we came up with was an approach to use our high-throughput experimental pipeline to take images of these animals throughout age. And then we used an approach called mask-RCNN, which is a convolutional neural network approach for image segmentation. And so we inject or we send raw images to this pipeline, and we first train the algorithm. So we're training the computer to learn what is a bead, a neuronal bead. We're specifically detecting these neuronal beads that show up through aging. And so first we train it, this is what this is showing, and then after we train it, we can just feed a new image, and the pipeline is going to first detect these beads and then quantify them. So how many there are, what's the neighborhood around them, how many beads are around, etc., things like that. And so just to show you how well this works, here on the bottom left, what you can see is that from images like this, once we send them through the pipeline, we can actually detect all the neuronal beads. Now, here it's very important to tell you why this is so difficult. So here on the right-hand side of this image, you see there are a lot of round, bright objects, but the majority of these objects are actually not beads that come from the neuron. The majority of these objects are what are just fat. So these are lipid droplets that these animals have in their intestine, and they turn out to be autofluorescent. So whenever you take an image of an animal, these little drops will show up, or lipid droplets will show up. And so the challenge here is how do we distinguish these lipid drops from these neuronal beads? And so as you can tell, this algorithm is pretty good at differentiating between those two, which is sort of the main challenge here. And so using this approach, I won't go into too much detail because of time, but using this approach, we basically, the first thing we did is characterize aging. So how do animals that go from here to here, what are the things that have changed? And here I'm just showing you one of the features, is the number of beats. As you can tell, they go up as animals age. Apparently what we see is that they increase up to day 6, and then after that, they sort of reach a saturation point where no more beats are generated, although we see other things changing. The other thing we realize is that if we put this neuron under stress, we such as cold shock, we also see some of these degeneration or beating happen, and might not be as evident as with aging, but it does happen as well. And here is quantified on the right-hand side, the number of beats and how they are induced with cold shock. And so in addition to this number of beats, once we have the segmented image, we can quantify many, many other things, such as their intensity, how many neighbors they have around, etc. And what we did, or what my student did, is he looked at 46 different metrics that where the goal was to construct a profile, a quantitative profile that can really tell us as much information about the patterns of these beads, the spatial patterns, as possible, right? And so by studying these 46 metrics, or some of these 46 metrics, the first thing we realize is it looks like the beating induced by cold shock and by aging is different. And this is a cartoon that sort of represents what we think is happening. And this is purely sort of a hypothesis we have that's supported by the metrics we've acquired. But basically it tells us that cold-induced beating does not perturb the dendrites, because what we see is more beats are generated, but their interbeat distance appears to not be affected. But in aging, what happens is more beats are generated, but they also get closer. And that tells us that maybe this disorganization of dendrites, which are generating dendrites in the center of the animal, is the reason for that. Now, this is, I want to emphasize that this is tricky. So here on the top, these images, one of these comes from aging and the other one comes from cold shock. And just from analyzing these images, it's really impossible to tell how these two are different. But the data does tell us that the degeneration used by these two are different. And to prove that, what Sahan did is he then used these 46 metrics to build a classification model. So these are models, some of these might be familiar for you. But these are models that include logistic regression, some machine learning approaches like SVMs or KNNs. And basically what these models do is they're first trained, just like the CNN we use for segmentation. They're first trained, we give them a few images and tell them, this set of images are aging, this set of images are cold shock. Now, if I give you a new image, are you able to tell me what it is? And so if you have good classification accuracy, that tells you that these two samples are quite different. And so we tried 4 different models, classification models, and Sahan was able to figure out that these neurodegeneration patterns are in fact quite different. It looks like I'm going to have to speed up a little bit. I might not have time to tell you about everything, but let me now switch to story number 2. And so I'm going to tell you about high-throughput screening now. The other very important thing we do in my lab to use these approaches is high-throughput screening, and or high-throughput genetic screening. So as I started my presentation, I was telling you how we're interested in connecting what genome is, or the sequence, the DNA sequences, to biological function. So basically what this means is, can we figure out the function of all genes? Because we know all of the genes that are out there, but we don't know their function. And so one way to do this is to perturb the function of genes and then characterize what happens. So you can knock out one gene at a time and then see what happens in the animal, for example. And so one way to do this is with what's called forward genetic screens. So in forward genetic screens, basically we start by saying, with a question of interest. And so here I'm just going to use a very simple example. So say we want to identify genes that are important for animal movement. Okay, so what we would do is we would knock out genes one at a time and then identify animals that can't move, right? And so then we could say, oh, these 7 genes, when we perturb them, they result in animals that cannot move. So that tells me these 7 genes are important in some way for animal movement. And so the way we do this is with a forward genetic screen is we take a population of animals and we randomly mutagenize them. So we put them swimming in this little chemical called EMS, which is a mutagen. It will induce random mutations in this population. And then we take this large population that we mutated and we search for animals that have the characteristics we're looking for. In this case, in this example of the animals can't move, or animal locomotion, we would be looking for animals that cannot move. We would isolate those and then generate more populations from them and study them separately. Then we could go back and see which gene was perturbed in each of these. So that's how forward genetic screens work in general. And so here, the first thing we did is we used an automated screening for synaptic establishment mutants. So basically, I've told you about these synaptic connections, right, a couple of times. Before, people had already run genetic screens to try to identify genes that, when we perturb them, disrupt these synaptic connections or the patterns of these synaptic connections. And here are a few examples. We call these easily identifiable mutants because, you know, they're easy in the sense that the location of these dots is completely perturbed. I'm highlighting in yellow where things are that shouldn't be there. But they're also not so easy because here each of these dots is anywhere from 1 to 2 microns. But now we were interested in looking at even more difficult things than those. And so we said we were going to look for subtle mutants. And so mutants that have changes that are more difficult to observe. For example, an increased number of dots, as we saw here. And so to do this, we use our integrated platform for screening. And this is where this video comes back. And so now I'm going to show you this video. Here's on the bottom left corner. This is what I would ask you to focus on. And what this is showing you is what the microscope is seeing. There's 2 boxes because we're doing 2-color fluorescence imaging at the same time. And what the system is doing is it's screening through animals in a completely automated fashion. The first thing it does is when an animal comes into the channel, it will trap it. It will determine if the animal is in the right orientation. And if it is, it's going to wait a little bit for it to cool down. And then it's going to let it go through one of the outlets based on the number of dots it has, for example. Now, why is orientation important? Because as you can tell here, these dots are only in the tail of the animal. So if an animal comes in with their head first, that's an animal we cannot analyze and we can just let it through. And so I'm having trouble with this. Let me see. Sorry, give me a second. Right here, let me just play it this way. So that was a head and it left. That was an animal that's in the right orientation, so it's going to be imaged, and you see the new picture appears here. That's also an animal in the correct orientation, so there's going to be a picture and then it's going to leave. And then this next, and some of the animals come in with their belly down, and animals that are tail first with their back down. But all of this analysis is done completely by the system. And so with that high-throughput screening, what we were able to obtain is a collection of over 20 mutants. And so what this graph is showing you is how drastically different they are from the wild-type animals. So the first colors, red, green, and yellow, all of those are animals that had been previously identified. And the ones on the right-hand side, all the blue ones, are the animals that we identified from this screen. It might be easier to look at the graph in the center. So this number tells you how different this animal is from wild type. And I'm going to go into detail into how we calculated this if anybody's interested. But basically, if it's 1, it's absolutely different from wild type. And the lower this number gets, the less different it is from wild type. And so here's just one example of an animal that we isolated 8 to 20. As you can see, 8 to 20 is over here. it has an area under the curve, this metric of 0.95 that tells us it's very, very different from wild type. But when you compare the images, they don't really look that different. So these are things that we cannot visually identify just by eye. So now that was one screen that we did. Now we're interested in doing high-throughput genetic screening for aging mutants. So instead of looking at genes that are important for establishing these connections in the right place, what we want to do is identify genes that result in a longevity change. So if you mutate this gene, the animal becomes or dies sooner, for example. And so my student Daniel Midkiff has been working on this, but I should first mention there's a few challenges, very significant challenges for doing high-throughput screening with aging mutants. And so, as I said before, animals live for about 21 days. So if you want to measure how long an animal lives, that's something that's going to take you 21 days instead of 3 seconds that you would spend taking an image of the synaptic patterns that I just mentioned. So it's a long experiment. We need to keep our animals age synchronized, as I mentioned before. You need to know how old that animal is, and for that you need to keep a population age synchronized. And the most tricky part of this is that to do forward genetic screens, what you need to do is when you isolate an animal, you need to make sure that an animal is still reproductively active, meaning it can still have babies, right? If that animal is not reproductively active, there's really no point in doing a genetic screen because we need that animal to reproduce so that we can study a large population of animals. Now, the problem is these animals stop producing progeny by day 5, even though they live 3 weeks. And so to do this, it's really tricky. And so the approach that my student Danny took is to use an early predictor for a lifespan, which is protein aggregation. So we based this off a previous study by the Della David lab, who showed that there's this protein called PAB1 in the animal that starts aggregating as animals get old. And if you label this protein with a fluorescent marker, you can actually see these clumps of protein appear in the head of the animal. And this is what you see here. Let me go back to the laser pointer. So this is what you see right here. Okay. So now our idea was, why don't we use this protein aggregation early in life, say at day 3 or 4, when our animals can still reproduce, and say whoever has an increased protein aggregation at that day will isolate that, thinking that that might tell us that these animals are going to live less because they're already showing higher protein aggregation than a healthy animal. And so that's exactly what Danny is doing, or he did. And what he did is he took this EMS approach where he mutagenizes a large population. Then he's using this filtering chamber that my other students hand developed. He cultures his animals there and removes all the progeny to keep the population asynchronized. And he does this until days 3 or 4, I believe. Then he sends these animals through the device, and he's able to isolate animals based on the levels of protein aggregation. And then after that, he grows populations, and he can quantify whether these animals actually have a shortened lifespan. And so here's an example of how this protein aggregation works. This is an original image, and using image processing, we can go from something like this to something like this, where we're just counting the dots. And as you can see, we can actually see there's a big jump from days 2 to day 6, which is just a validation that our system is working for detecting this protein aggregation. So Danny actually went through this process, and he screened some longevity mutants. He has so far screened through 1,900 animals. And after looking at those, we have over 7. He's still working through this, but he has over 7 mutants that are aggregation mutants, meaning they have higher levels of aggregation. And at least 2 of these we have seen that are aging mutants. So as you can see here, this is, and I forgot to mention this is a survival curve. And so this tells you how many animals are alive as time goes by, right? And so the average lifespan would be 50%, right? That's when half of the population is still alive. So for a wild type, animals start to die off maybe at day 5, and then by day 20, they have all died. And for these 2 specific mutants that he has looked at, they're all dead by days maybe 13 and 16, but they also start dying sooner. And so here's an example, or here's the data for the total aggregation that comes from the pharynx. I think in interest of time, I might just tell you about this real quick. So just super quick, this is a third story I was going to tell you about. But basically what we're interested in here is, can we predict how long an animal is going to live? And the way we're doing that is we're using this key gene called DAF16. And so DAF16 is a gene that's been known to regulate aging. It's the master regulator of aging. And what we're trying to do is to track throughout the animal's entire life how active this gene is. But that's difficult because if you want to track the activity of a gene, current methods, the only way you can do that is to sacrifice your animals. So there's no way you can track them through their entire life. And so what we are doing right now, Javier, another of my grad students, is he used a CRISPR approach, and hopefully you've all heard about CRISPR, but if not, that's okay. CRISPR is just a sort of novel way to perform genetic engineering, so to chop and crop and copy and paste pieces of DNA. And what we're doing is we're using this CRISPR approach to fluorescently label the DAF16 gene in this animal. The important thing here is that we are tracking the endogenous levels of gene. We're not tracking some artificial array that's external, but here we're just tracking exactly what the animal is doing with a fluorescent marker. And so just to show you how this looks like, you see that this gene is showing up in the hypodermis. All these little cells you see here, those are the ones that have DAF16. We also see it in the intestine, as you see here. Now, why do we have 2 rows? The top row it says 0 hours, and the bottom row says 12 hours. These are hours of dietary restriction or fasting. And it's well known that DAF16, this gene, responds to fasting. So when the animals go into starvation, this gene gets activated, and we actually do see increases mainly in the neurons. All of these cells you see here in the head of the animal are neurons that we see light up once we make these animals fast. And so, long story short, what Javier is doing is he's quantifying this amount of DAF16 intensity using image processing approaches. And he's also doing different treatments to the animals, different starvation treatments. So we give them different amounts of food so that we can get different amounts of DAF16. And then he's also quantifying lifespan. And we can use a device like this to do the high resolution imaging of many animals at once. And just to sort of the big conclusion of this project thus far is that we can very reliably correlate how long an animal is going to live with how much total activity this gene has been under for their entire life. Now, this is just comparing the lifespan with the total activity of the gene, and the way we quantify this total activity is we just add up the entire intensity we get from our images for all the days. But this might not be the most relevant thing to look at. So what we're doing right now is we're trying to figure out if the intensity, for example, in one of the tissues is more important, or the activity in one of the tissues is more important, or is it the activity of DAF16 in their larval stages, which would be like childhood, or is it in the middle age, or is it later, or is it all important? And so for that, we're really currently going through a lot of data analysis. And I'm going to skip that because I think I'm running out of time. I would like to thank you all that joined for listening to me. This is work that could not be possible without my amazing grad students that are here and with their funding sources. And if anybody has any questions, I would be more than happy to take them now. Thank you. Oh, okay. Yeah, that's all right. It's okay. Oh, that's a very good question. Yeah, so basically, let me go back. So this was not so easy. What my student did is he took a bunch of images from this, and then he used a code in MATLAB that lets you draw around the beads. And so we use the pad that you can use to sort of with a little stylus to draw around the beads. And so we just built a training set that way. We're aware that that's not completely accurate because these animals have anywhere from 30 to 150 beats. So it's very easy to make a mistake or to mislabel things, but it still worked. And actually, what we realized is that after we ran the CNN, when we compared our training set, we could visually see that the CNN was able to find some beats that we didn't initially label because it was hard. But yeah, that's how we did it. Yeah, yeah. Yeah. Mhm. How many what? Sorry, can you repeat? Oh, the valves. Yeah, yeah, yeah. So I think the device that we've had with most valves is something like 16. Um, and I think that it depends how um you organize. Usually the problem with valves is that you start getting channels that cross each other. So the microfluidic device design is what makes it difficult. But the what we're using here are single layer, these are called single layer valves because the valves are on the same seal in the same location as the channel, so they close from the sides. But you can also do 2D valves where instead of doing that you put the valves on top of the channel and you close from the top. And so if you do that, you don't have a problem with channels crossing each other. And that way you can have many more valves, as many of pressure sources that you have, basically. And so right now, I think our valve box has up to 16, but you can easily, and there's open sources places where you can construct your own pressure box, but you can build up to, I don't know, 64 or more. So, yeah. Mhm. Right, right. Okay. Sure. Single molecule? I have not. Yeah, I think it's I mean, I think that would be interesting. Um, but I haven't thought of it maybe just because it's not my expertise, but it would be, yeah, it would be interesting to do that. Yeah. Yeah. Thank you so much. Okay, perfect.